Several of you ask about nuclear pores. Now, just as a reminder, the nucleus, and some cells do have more than one nucleus, but most of them just have one. And the nucleus has a fluid inside called the nucleoplasm. Floating around in that fluid would be one or more nucleoli. If it's just one, it's a nucleolus. And this is where the ribosome subunits are put together. Then there are the chromosomes that are throughout there. And yes, the nuclear pores, the bigger holes. So those are holes that are there so that messenger RNA, which is a product of transcription, can get out of the nucleus. It does require some energy, by the way, because there's more messenger RNA being produced in the nucleus than outside in the cytoplasm. The messenger RNA carries the information from the DNA for putting a protein together. It carries the recipe, if you will. We'll talk about that later on in the quarter. The DNA, though, also transcribes ribosomal RNA that will go to the nucleolus and then bind with proteins to make the small or the large ribosomal subunits. They go through those nuclear pores. And there are some genes on the chromosomes that make what are called transfer RNAs. And those need to come out the nuclear pores. These are relatively large macromolecules or structures that need a bigger hole to go through. And because they're made in the nucleus, it's active transport because, that's actually high to low. But I think it's because they're so big. I was reading that they need energy for this to happen. That's what active is. Okay, sorry about that little mess up. But now hopefully you all understand that bigger macromolecules or structures move through those nuclear pores. There are, simple diffusion and facilitated. It's almost like a special type of facilitated diffusion with a little bit of active transport added to it. But don't worry about that. I'm not going to ask you about that as far as movement with the nuclear pores. Okay, I think that was everything people wanted to know about the nuclear pores. Now, I'm looking at my notes here. This turned out to be harder than I thought to collect all these questions. Okay. Then someone asked about smooth versus rough endoplasmic reticulum. And they're both just these membranous tunnels, kind of flattened. The smooth endoplasmic reticulum does look different in two ways. It's not as windy and flat as the rough endoplasmic reticulum, and it doesn't have ribosomes. That's what all these little dots are. Now, that doesn't mean that there aren't ribosomes that are free ribosomes and out in the cytoplasm, because this is outside the nucleus. But the rough endoplasmic reticulum, because the ribosomes are attached to it in a eukaryotic cell, is where protein synthesis takes place. We're going to learn more about that later in the quarter. So you might be thinking, well, what about a prokaryote? It doesn't have endoplasmic reticulum. Of course it doesn't. It doesn't have membrane-bound organelles. But it does have ribosomes, free ribosomes, and that is where the protein synthesis takes place in a prokaryote. The smooth endoplasmic reticulum is specifically for lipid synthesis, not protein synthesis. So you do not see these ribosomes on the surface of the membranes. All right? And that's all you need to know about those. But I will tell you, it relates to the Golgi, which also relates to vesicles. And there were quite a few questions about vesicles. So what will happen is immature proteins will be formed. They won't be completely mature proteins necessarily. Some will, some won't. And some of them are moved to the Golgi, where they are packaged in vesicles, specifically lysosomes and peroxisomes. Oh, I wrote it down for vesicles types. Synaptic vesicles. We don't have to get into that. If you take anatomy and physiology, you will get into that. But what happens is there are proteins that need to be put in vesicles. And that's what the Golgi does. And so it makes all these different types of vesicles. There are lots of different types of vesicles. What in general defines what the vesicle is, the type, is what is inside it. So lysosomes and peroxisomes contain hydrolytic enzymes. Enzymes that digest bacteria, for example. So your white blood cells run around, bacteria might get into your blood, and they engulf that, make a food vacuole, which is also called an endosome. And it's an early endosome when it first engulfs whatever it is from outside, and then it matures and becomes a late endosome, and then fuses with a lysosome or a peroxisome, and the contents, those hydrolytic enzymes that are proteins, go inside and eat up the bacterium that's in there. And hey, that helps you not be sick. Woohoo! Okay, so different types of vesicles depending on what's inside them. Smooth endoplasmic reticulum, remember, lipid or fat synthesis, some kind of lipid. They can move to the Golgi and also complete their maturation and maybe eat. I don't think many of them are in vesicles, but they complete their maturation process. So the Golgi is for completing maturation of proteins and lipids and for packaging in specifically those proteins. Okay, vacuoles. Vacuoles are really big vesicles that aren't made by the Golgi. And not all cells have them. I gave you two examples. Plant cells. In general, a living plant cell has a huge vacuole that's a water vacuole. Everything else is pushed to the side around that membrane structure that's holding all that water. And you know what that does? That keeps your plant nice and healthy and standing straight up. If you don't put water in the pot with the plant, it will start to wilt. That is because those central vacuoles are losing water, and they can't keep it's a turgid thing. It can't keep the structure of the cells, and then collectively all the cells can't keep the structure of the plant, so it wilts. We have fat cells, for example, that store fat. Fat has different lipids that can be used to make ATP. Again, we're not there yet. But that's for energy storage and for insulation and for keeping some structures in place. Okay, so, and I already said that prokaryotes don't have vacuoles. They have areas where materials are concentrated instead of vacuoles because they don't have membranes. Okay. Mitochondria and chloroplast. There's a mitochondrion. And here's a chloroplast. And there's a double membrane. So, this is a chloroplast. And we're going to talk about these in more detail later because they are involved in energy transfer. So autotrophs, or photosynthetic autotrophs, have chloroplast. And chloroplast are the sites of photosynthesis. So light comes in, and along these membranes, the light is captured, it excites electrons, it makes these compounds that go to what's called the Calvin cycle that's inside, also inside the chloroplast, and it makes glucose. Okay? That's what they do. The product of photosynthesis is glucose. It's actually half a glucose, and then those are put together. But anyway, that's what that's about. Okay? What happens then is either the photosynthetic autotrophs, to run their cells, they need ATP, we need ATP, so plant cells, for example, have both mitochondria and chloroplast. We don't have chloroplast, so we have to eat some kind of autotroph or something that ate an autotroph, or something that ate an autotroph, depending on, you know, how our diets are arranged. But the mitochondria is where the glucose, the energy in the glucose, is converted into energy in ATP that we use. And again, we will talk more about ATP later. Nearly at the end, cell walls. Someone said, hey, why do some plant cells need 2 cell walls? Well, because they function more in the structure or the movement, specifically of water in that plant. So they need to be very rigid. So each cell wall provides rigidity. 2 cell walls even more so. Why don't animal cells have cell walls? Well, they didn't evolve in animal cells. And if you think about how materials are moved throughout an animal cell, we don't need cell walls. In fact, they would be a problem for us, the way things are moved. So it's structurally incompatible with an animal cell to have a cell wall. And then, finally, someone asked about locomotor and non-locomotor appendages. So basically, appendages are just something that stick off of the cell. Locomotor appendages are ones used for locomotion or movement, like cilia and flagella. Okay, so the cilia are shorter and more numerous. Flagella are longer, like a sperm cell has 1 flagellum, and it helps it swim. Okay? So it can maybe get to the egg. All right? The non-locomotor appendages are not for movement. So bacterial cells, for example, have what are called fimbriae. You don't need to know that, but they do have these appendages that allow them to stick to host cells so that they can cause all the havoc that they might cause if the immune system doesn't stop them before they do, or you don't take a drug to stop the bacteria. Bacteria also use what are called pili. These are a different type of appendage to swap pieces of DNA. So non-locomotor appendages have other functions. You don't have to worry so much about that for this class, because really, cell biology is huge. Cell biology for majors takes up a whole quarter. There is so much more depth to cell biology that all we can do is look at some of the structures and then look at some of the particular functions. So now we've talked about cells, we've talked about their components. I want to make clear that both prokaryotes and eukaryotes have cell membranes. Both prokaryotes and eukaryotes have ribosomes. Both prokaryotes and eukaryotes have chromosomes. So they both have cytoplasm. They both have cytoskeletons, but they're different and more involved in the eukaryotes. But what they don't have What prokaryotes don't have are all those membrane-bound organelles that you've got a whole bunch of them in the notes that eukaryotes have. All right, so that is the end of the second and last lecture for this week. So thank you for your questions. Thank you for clarifications. And now you see how this is going to work. Each week, you send me the questions for the new notes or the clarification request, and I do my best to cover those at least to the level of what I'm going to quiz you over. All right, bye.